Howdy, my name is Ethan Fritchie, and my project assesses how specific genetic traits of the honeybee allow for resistance against varroa mites. Apis mellifera, also known as the honeybee, is a winged organism that lives in huge colonies inside of a hive. They collect pollen and nectar from a bunch of different plants and flowers and use it to produce wax and honey. It's estimated that these social insects pollinate around 4/5 of the world's plants, including 90 different types of food crops. This is why they are so essential to our ecosystems and to world food production as a whole. In recent years, however, their population numbers have been on the decline. One of these reasons is due to parasitic mites. Varroa mites, specifically the Varroa destructor, are one of the greatest threats to both wild and managed honeybee colonies in the world. They directly impact honeybee health and transmit a wide variety of devastating RNA viruses, most notably the deformed wing virus, that spread within and across colonies like wildfire. This is the reason that researchers have begun analyzing the science behind protecting these honeybee colonies through breeding them for specific genes. So, my question is, what genetic traits allow honeybees to tolerate and resist parasitic mites and the viruses that they transmit? The answer to this question is the key for effective breeding programs which look to reduce the risk of these colonies being affected by varroa mites. I took a look at 3 different experiments to assess this question. The first evaluated the amount of natural virus infection and immune gene expression in 2 different groups of colonies of bees. These colonies were either classified as R, resistant to mites, or S, susceptible to mites prior to the sampling, with the R colony having survived without chemical treatment for more than 2 years, and the S colonies coming from a population known to be vulnerable to varroa. They used quantitative PCR to characterize differences between the 15 R colonies and 15 S colonies. The results showed that R bees showed much lower natural levels of deformed wing virus, and also decreased levels of black queen cell virus and cashmere bee virus than the S bees did. The S susceptible bees, on the other hand, showed much higher amounts of cellular immunity genes, EATR and NIM2C, than the R bees did. Surprisingly, resistant bees showed higher levels of gene encoding the antimicrobial peptide hymenoptysine, and this was a major driving force in separating the 2 colonies by cluster analysis. This cluster analysis diagram shows the results that we talked about in the previous slide. It shows a much higher virus level in S bees than in R bees, as well as higher levels of hymenoptysine in R bees and the NIM2C and EATR genes in the S bees. The next experiment that we looked at again classified the R and S colonies prior to sampling. Total RNA was then extracted from the individual bees. RNA sequencing data for gene expression differences and virus levels helped us to differentiate mite-infested bees of the R and S genetic backgrounds from the sister bees that were also studied and were mite-free. The results showed that RNA sequencing data revealed very noticeable transcriptional differences between mite-infested and uninfested honeybees, and between R and S phenotypes. First, the R population didn't show an increase in viral levels with the varroa presence. Second, in the susceptible S colonies, mite-parasitized bees had significantly higher viral loads. Both mite-free and parasitized bees had much lower mean viral levels than the mite-free and mite-parasitized susceptible bees. In total, 847 honeybee genes were shown at higher levels in S bees with mites than in R bees with mites. This graph gives supporting evidence to the results found in Experiment 2, as we can see that the viral levels were much higher in S bees than in R bees, regardless of whether they were mite-infested or mite-free. In our next experiment, varroa-free bee pupae were injected in a laboratory with the deformed wing virus or a phosphate buffer solution. This time, the R and S phenotypes were characterized by the DWV copy number after they were injected in the pupae. All of the colonies in this experiment were also mite-free. They then collected RNA for sequencing. The results showed strong contrasts when looking at the responses to the virus injection and the R and S genetic backgrounds, as well as gene expression when comparing the response to the BPS injection. Both the identity of the genes being expressed in the experiment and the direction of the expression level change, which could be either elevated or reduced, differed greatly among the mite-free bees drawn from the R and the S colonies. In this graph, we see this great variance in results, as pupae from some colonies showed high viral loads with either condition, which could suggest that some with already high amounts of viruses were just amplified even more regardless of the new additions, while others showed significant increase in viral loads when the bees were injected with the live virus. Two sources also had relatively low average amounts of virus, with both control and live virus injections. In conclusion, families of bees with lineages that had naturally low levels of the DWV and higher levels of the gene, which encodes the hymenoptetasen, tend to be mite resistant. Also, genetic changes that differentiate these R and S bees affected by Varroa and DWV, especially gene expression alterations, may be the key to enhanced viral and Varroa mite resistance of the R bees. Finally, the identity and direction of the gene's expressive level can have a great impact on whether or not a colony is resistant or susceptible to the Varroa mites. In recent years, the population of honeybees have been severely impacted by Varroa mites, more specifically the Varroa destroyer. These parasites are the single most prominent threat to both wild and managed honeybee colonies in most of the world. Continuous chemical use is not a long-term option, however, as it can lead to resistance by these mites as well as contaminated products. So, discovering what the genetic makeup of a mite-resistant honeybee consists of has become of vital importance. This way, we can continue to breed generations of healthy, mite and viral-resistant honeybees that will continue to serve our crops and ecosystems as a whole. Thank you.